everyone and welcome. My name is Selvi and I'm account manager at Novagene Europe. I will be your host for today's webinar. And thank you so much for joining us. I am delighted we have participated from various backgrounds with us today. In this session, we honored to have Dr. Maria, who will share insights on decoding with soil microbiome introductions, shotgun metagenomics of Rhizosphere soil across UK agricultural farms. Before that, I'll take just a few moments to introduce you to Dr. Maria and our company. If you are ready, let's get started. So, as I said, I like to introduce Dr. Maria as a senior research fellow at the John Innes Center in Norwich, where she has been working since 2019. Her expertise lies in plant soil microbiome introductions with a strong focus on the Rhizosphere, microbiome, and nitrogen and carbon cycling in soils. So Dr. Maria has also coordinates the international Wheat Roots, which is EJP soil projects, and is a recognized leader in biological nitrification inhibition and functional phenotyping approach. So importantly, she works closely with farmer networks, breeding companies, and global research partners, helping to translate research into the real world impact. So before I like to talk about these research life offers, which is a global exposite through live webinars and expert question and answer session. So it is a space to discover and exchange innovations and to collaborate across the borders with leading scientists in the field. Many of you already know maybe, but I like to also talk about to our company, as I mentioned before, and Novagene was founded in 2011 and has been growing rapidly since then. We are constantly developing our portfolio of NGS and third generation sequencing. And today we are present with services in various fields of science, like from research to food and water testing and oncology. And we have like 10 laboratories around the world, and our business covers around 90 countries and across, again, 6 continents. So our first European sequencing center is in Cambridge Science Park in the UK. And the UK lab is equipped with Illumina technology and our patented intelligent sequencing platform, Falcon 3. And this fully automated system integrates multiple precision intermediates to streamline DNA and RNA sequencing workflows and significantly reducing the turnaround times and minimizing human error. And Falcon 3 ensures contamination-free and high-quality results and enabling us to process large volumes of data efficiently and constantly. And in addition, we have another NGS generation sequencing lab in Munich, Germany. The Munich facility is rapidly expanding its operations and is equipped with both NovaSeq X Plus and PacBio Revio sequencing platforms. So also we have a new machine, which is Elements sequencing platform with there. So which means the future enhancing our capacity to deliver the cutting-edge genomic solution across to Europe. So in here, you are seeing our sequencing services, which is unlock the full potential of your research with our comprehensive sequencing services from the human genome to transcriptome, microbial genomes, and epigenomes. And so we deliver, again, as I say, cutting edge insights tailored to your needs. So also explore the complexities of animal and the plant genomes with our advanced platforms and dive deep into proteomics to understand protein dynamics. So yeah, powered by the latest technologies, and our services offer precision, speed, and reliability, and fueling discoveries that drive science forward. To join the pioneers benefiting from the world-class genomics and proteomics analysis today. So maybe you can ask, maybe you can ask why you should know a gene. So obviously, choosing Novogene as your sequencing partner means choosing unwavering quality and reliability. So we prioritize excellence through the rigorous quality control at every stage. So sample, library, and data QC to ensure your results are accurate and trustworthy. Our growing capacity, so boosted by the new, like our German lab and our patent Falcon 3 technology. So not only enhance data quality, but also speed up turnaround times, which is our best power, and also running like simply 20 hours and 7 days. We also offer flexible end-to-end solution tailored to your needs, whether you require the full project management from like extraction to analyze data, or simply need raw data from supplied DNA, RNA, or libraries. We are offering also pre-made libraries services as well. So we adapt to fit your workflow. So our dedicated product management team and expert labs constantly optimize the protocols so to deliver the perfect sequencing solution for you. Yes, we are actually as a like Norwegian and in Europe. So yeah, contact is always close by. So with local representative who speak your language, and we are here to support you every step of the way. Yes, let us help you achieve your research goals, and with like custom service and expert guides tailored to your needs. So thank you for your attention. So that concludes my part for now, and I'm pleased to hand over the session Dr. Maria. Also, in addition, I like to say we will have a poll question. We would like to have if you vote. And yeah, Dr. Maria, please take it away. Thank you.
Thank you, Selvi. Okay, let's. Well, good morning, everybody. Thank you for joining us this morning for the webinar and for your interest in our work. And well, Selvi has already introduced me very nicely, so we can escape that. And as you can see, today I'm going to be talking about the interaction between wheat and soil microbiome. And particularly, I'm going to be presenting results from a project called Truth, in which we are examining the impact of different soil types and soil management in real farms. And we are supported by Novogene into doing our microbiome analysis. And then I will also, at the end, I will explain how the Truth project is also linked to the Wheat-Microbe projects that Selvi has already mentioned. So the Truth project is mainly this team, and though there is more people always behind the scenes, this is the main team involved in this project. So it's led by BOFI, which is a British on-farm innovation network, which is led by Tom Allen Stephen. It's a big community of over 300-400 farmers all over UK that are interested in improving the quality of their soils. Then we are supported by Agritech in terms of management, data processing, and reporting to the farmers. At John Innes, we are running the microbiome analysis of the samples that the root rangers collect themselves. You will see that they are doing some soil structure analysis in the University of Nottingham. And all the data that we are collecting and processing, we are with best technologies is the company within the project. They are going to be using it to develop this sensor that, according to them, can smell the soil and provide analysis of the main soil health parameters. So this is at the website of the project. I invite you to visit it if you want to know more about it. As you can see, it's running for 3 years. We are entering the last season. And the results I'm going to present you today are from the 24. Since the 25, we are still processing samples and all the data that is being generated. Also, if you want to get in touch with farmers and the community, there is a WhatsApp and a platform where you can join the Soil Circle, where people are actively discussing all sorts of issues with soil health, particularly in farms. So we call our farmers that are enrolled in the project that actually take the samples and send the samples to all the academic partners. So we call them root rangers, because the project is obviously focused on the impact of roots in soil health, both control of microbiome and impact on soil structure and mycorrhiza fungi. So in 2024, 10 root rangers were recruited all over UK. You see there are very different locations. And with different locations comes something very important, particularly in UK where the soil is quite patchy. You see that pretty much every farm, except for these 3, will have a different type of soil. And that's going to have a tremendous impact in the microbiome functioning and composition. Besides the soil classification in terms of sand, silt, clay, etc., we also have it classified in what kind of land management the farmer is doing, whether it is organic, regenerative, or conventional. So organic is a well-defined, reasonably well-defined boxes that you have to tick to actually declare your farm organic. So it's more it's more easy to integrate it as a parameter. Regenerative is, as I will show in my last slide, it can be a mix of many different things. It can be just one approach. It's just a transition between the most conventional ways of farming to something that is more focused on decreasing inputs, etc., to improve soil quality. So our sampling strategy for 2024 in terms of collecting soil for microbiome analysis was collecting samples before fertilizer was applied, in case it was applied. Obviously, for organic, they will be applying other products, and then collect a second set of mirroring samples after the fertilizer application to see and also evaluate the impact. And in this first year, we took, as a control, we took a number of bare soil samples that were not impacted by the wheat. May I say that all these farms were growing wheat, a variety of modern wheat. Then I also was curious to examine a bit how the margin, where no intensive labor in the farm is doing, how the microbiome will be looking. And finally, of course, my focus was the Rhizosphere to see this impact of different wheat with different soils and different land management in the microbiome. And then we did a lot of analysis. So obviously, supported with Novogene, we did the shotgun metagenomics of over 200 samples in the first year. And then not only the taxonomy we measured, but we could also predict KEGG models and KEGG genes, which I'm going to explain in a second what it is for those of you that might not be super familiar with it. And we could also predict Cas-symes. The interest of this is like beyond having a library of names of bacteria and fungi, etc., of microbes, we can actually have a look at what functions potentially they can have in the soil. And we supported all this microbiome analysis with basic measurements of soil chemistry, nitrification potential in the soil, soil structure by Nottingham, and mycorrhiza. So this is very quickly what is the workflow. So farmers will collect the soil samples that I have just explained. Here at John Innes, I will perform the DNA extraction with just a conventional kit. And then at Quadram Institute is also our neighbor building in the research park. They help us with the library preparation. If you want more information on the protocol that it's followed there, you can visit the website, and the protocol is available for free to have a look. And obviously, then it comes one of the most important bits, which is sending the samples to Novogene and have the sequencing. Here you have the details NovaSeq Plus. I think Selby could answer any question you might have about what is the advantages of using this platform. And then Noviem will provide me with the raw data. And go back to Quadram. I work with some colleagues that have a very specialized pipeline that is very helpful, particularly with very complex samples like soil, where there is a lot of noise and many thousands of different species to organize. So I'm very lucky to have these colleagues that are happy to collaborate with me. And then once the raw pipeline has been, the data comes back to me, and I start doing statistical analysis and interpretation. So I mentioned that we do some supporting functional assays like the nitrification rate. I just wanted to mention, so we collect the soil, we incubate it in specific conditions, adding some known amount of ammonium sulfide. Then we are very lucky that this is high throughput because we can measure nitrification over a week using just a probe that has been developed by this company in UK. And we just measure the slope, how quickly the ammonia is transformed into nitrate. Why is that interesting? Well, nitrification is something that many of you probably, if you are familiar with my work, or even if there are many experts in ammonium, in nitrification, inhibition, and so on. Nitrification is, you cannot say it's good or bad, it just depends. So that's why I have highlighted here. So again, if any of you are not familiar, I have dropped here this basic scheme on how ammonium is a form that the plants can take. Somebody sometimes apply directly from the fertilizer. There are many communities in the soil that we use it to gain energy. So they will transform it very quickly into nitrate. Then other communities will complete the job to nitrate. Nitrate is very soluble, but the plant will have to transform it again back to ammonium to actually be capable of processing it for proteins, etc. But the main problem is not that. Most plants, most crops are happy to have a mixture of both. So it's not something we want to completely stop. But it is true that nitrate has more risk of leaching and transformation into greenhouse gases like nitrous oxide. So I leave you here with a list on, yes, it's good. It supports it's part of the natural cycling of nitrogen in the soil, and it supports many microbes in the soil that obviously are also linked to other microbes, as I will talk in a sec. So it completely suppressing nitrification will be a big disruption of how the community of the microbiome works. The negatives, I have already gone through them, obviously losses of fertilizer, economical losses, and impact in the environment. So as everything in life, the important thing is to find an equilibrium. This is as one slide to show you how the soil structure is being assessed for the Truth project. This is the work they are doing in Nottingham, and the number of parameters that they provide to the project and to the farmers to assess a bit the porosity, particularly a number of volume of soil pores, is something that me as a soil type scientist interests me a lot, because it will create habitats for the microbes, and it will have a direct relationship with water holding capacity, which is very important for soil health and for the health of the microbiome. And finally, Tom in the University of Cambridge, in the Crop Science Center, he's contributing with the analysis of microscopic analysis of arbuscular and mycorrhizal fungi. So all of this, I have said, this is as the website of PES if you are interested in the instrument they are developing, and it's all the data that we are producing. They are doing some machine learning training to get the PES the sensor to provide as much information as possible. So going back to that, I was talking that in the 24 we collected soil from 3 different compartments. We are going to call them bare soil, rhizosphere, and field, and from 3 different land management. And we also have the different soil types. So that's the 3 main variables that we are going to be discussing. Despite the fact that, as I'm going to show in my results, soil type is the main driver of the microbiome composition and all the other things that we have been measuring, there is also a contribution of the land management, and despite the reasonably low number of farms in this previous year, there is still a significant contribution. And for that, I'm actually going to focus on the management. So why did we decide to have 3 compartments? If you have never seen an image like that, this is it's a quite visual aid. Bulk soil will have a very different rhizosphere environment, microbiome environment than the rhizosphere. Basically, we will have higher diversity in the bulk soil, but less diversity in the rhizosphere because it will be enriched in microbes that the plant is actually interested to interact with. So it will be a much more dynamic activity because there will be this exchange of compounds from the root to the microbes and vice versa in some case. The bulk soil is a much more stable community, so this is more dynamic. And you will definitely find in the rhizosphere an enrichment of several taxa. I'm not going to go so much today into which taxa is there because it's quite robust. It's something that we see repeatedly across soils, which are the dominant taxa that are there. So I think what I wanted to present you today was more about like what can they be doing. Let's remember that what I'm going to present is potential functions. And I thought that will give you a much more interesting idea of how the different land management and the soils are working. So I was saying we have the combination of different type of soils, different land management. This is just a bit of a cartoon showing you the complexity that adds to the question. It's like, so when you ask, how is my soil doing? Your soil, it's the microbiome activity in your soil is going to be, particularly if you look at the rhizosphere, which is my main interest, it will depend. You can see that a sandy soil with organic management will differ different than a clay soil with organic management and all the different comparisons. So the sandy soil will be the main driver. So that's why it's more highlighted. So we will have, because of what I was talking about, the soil structure, porosity, and so on, in this sandy soil where there is much less structure, the microbes are going to be more dispersed. In a clayey soil, we are going to have more aggregation because structurally, so that's going to provide these microhabitats. So that it's going to determine the recruitment that the plant can do. Is going to depend very much in this texture or this structure of the soil. Now, obviously, organic and conventional as management effects compared, so they will also have an impact on the main microbes that are there. But again, as you can see, that's going to be a balance between what's happening and what is the environment that they are living in as well. So I hope that's interesting, and it sets you a bit in the mood of we need to consider these 2 parameters. So like I say, I'm not going to go into the results of the truth for the taxonomy because basically I just wanted to give you a flavor. Again, if you are not familiar with what's in on what's a normal microbiome profile in an agricultural soil. So this study was published a few years ago already, showing something very parallel to what we are trying to study in the farms. So you can see here the main players. Obviously, this is at the phylum level, so it's very general. At this level of taxonomy, within each phylum, you're going to have very different actors, but it's a very broad kind of a stroke in what are some of the important communities that we want to look at changing and interaction when we are looking at the impact of land management in an agricultural soil. With that said, I was telling you that about the KEGG functions, KEGG genes, KEGG modules, and Cas-signs. Again, just a little bit of an informative slide. What's the difference? So, well, we can just look at the genes. So from the taxonomy, we can predict which functions are being carried, which genes these microbes are carrying. Looking at genes individually can be a bit tedious and perhaps less informative. So the next step is to look at the modules, which are giving us an idea of an actual pathway, because sometimes we see long lists of KEGG genes that will be having very, very general functions. But if we can put all these genes together within a pathway, then we can talk more about an actual function in terms of if it's a nitrification. Saying that we have a transporter might not have that much meaning on its own, but if that transporter is within a particular pathway, nitrification, carbon degradation, etc., or many other actually. So that actually is like a glycolysis, for instance, that's something more informative for us. And well, I just leave you here for if you're not familiar with the PEG is. It says the Kyoto gene classification. So it's a free resource for anybody that is in need of using it. So with that, I leave you that the KEG modules is what I'm going to be presenting some results today, because obviously I'm interested in particular actual pathways that can be affected by the land management. And the cazymes, but they are carbohydrate active binding modules, so just cazymes for short. And within that similar approach, a little bit to what I was explaining with the KEG modules, we want to look at full functions, not just a very, very specific, but something that can have a meaning in terms of how my carbon is going to be transformed in the soil. So you see, it's quite a complex picture, because carbon turnover in soil depends on many things. It will depend on different sources that are entering the soil, whether they are very labile or they are very recalcitrant. So like cellulose or lignin are going to require a lot of more time to be degraded, and also more specific enzymes. That is, it's not like glycolysis or glucolysis can help with very labile carbon that will be degraded very soon. This will require a much more complex set of enzymes. So you will have also cazymes released by different microbiomes, and obviously there is also an input of these enzymes from the plant. And finally, today I'm not gonna present results about the network, because we actually, despite the fact that we have over 200 samples, we are slightly limited still to create networks that we can actually compare. Creating networks is really, really interesting, because as you can see, it allows us not just to see isolated microbes and how they are going up and down, but actually how that reflects in the entire community. Obviously, they interact. So you can see here the green interactions and the blue interactions. That will mean there is either a positive interaction and negative. So when one goes up, the other one goes down. Or some of them will not. It will just be kind of in a balance. But those interactions are really, really important for the functioning of the soil. Okay, but some results now that I have given you a good overview of what you're going to see. In this first slide, so we have the farms coded by farm 1 to farm 10, because obviously we want to respect a bit the privacy of our root rangers. But as you might, they are still color coded in this case, by the management. So that's why they are ordered in that way. If we look at the basic microbial diversity, is a very standard parameter to look at soil microbiome, but we can see that it remains very comparable across the farms. So we are not going to see much in that sense. Most of the farms are bacteria dominated, which is something we can expect from agricultural soil. One thing that was interesting is the nitrogen cycling potential did vary between farms and between management types, which is telling us that different fertilizer supply, different management, etc., is going to have an impact in the nitrogen cycling that I was showing you before, which is one of the main focus of my research, together with carbon. So you will think carbon degradation is enhanced in regenerative and organic soil versus conventional. Is that a bad thing? Not necessarily, because the turnover of carbon that is a positive for the release of nutrients that are naturally stored in the soil and the plant will need. So that turnover of carbon, that degradation, is actually positive. Like everything else, it has to be balanced. And we found, as you can see here, that the potential nitrification rate that we measured at Unioness, we found that it was quite high for most of the sample in some of the farms under conventional, which is also I was expecting in the first place. And then, oops, sorry. Then I wanted to also present you the same results, the same diversity, some of the genes for transformation of nitrogen and carbon degradation and infection rate by the soil. Obviously, I'm very constrained. It's very difficult to compare here because of the number of soils that we have per type of soil. So that's something that we are going to probably expand this year, because in 25 we have 20 farms, which has duplicated the number of data. But we can still see some differences expected by the type of soil. So that's a very basic overview of classic isolated parameters that we can measure, not just the diversity or the fungal bacteria ratio, but also some more in detail potential functions of the microbiome that are being affected and functional outcomes. So what we are looking here in terms of the beta diversity, I'm going to draw your attention first to the panel D, because here is what I was mentioning at the beginning when I presented you the different things. The soil type is the main driver of diversity. It's the one that explains the more variation in our microbiome composition, but also obviously in our microbiome potential functions and caseins, etc. But still, it's good to see, as expected, the compartment, which was bare soil, margin, and rhizosphere, it also has an effect. The timing, remember we measured before and after the fertilizer, didn't seem to have any impact. So the good thing is that then we can use that as a random variable and we can integrate all the data. So that duplicates our number of replicates. But I was quite pleased that at least the management shows a significant impact. So here you can see differences in beta diversity for soil type and differences in beta diversity for management. And we did have, it's quite fun to see, because if you look, conventional is right in the middle. Organic was actually significantly different, and regenerative, some of the farms are closer to the organic, some of the farms are the conventional, because as I was explaining, it's a transition. And we also had some significant differences also in the compartment, as it's summarized here. So before I take you to the next slide, just a little break, so to speak. There is one variable that you probably have in mind that I haven't talked about because, but two actually. If you remember the map, these farms were spread over UK. So we are talking about different latitudes. So we are going to have, we can expect different impact of the climate from the northern farms to the more southern ones. So that's one. And the other one that we are going to be having a tremendous challenge when we want to compare 24, 25, and most likely 26, is the dramatic changes in weather condition, because when I go to the field every year seems to be a completely, it's like doing my field trials in a different country. So I pulled together some data that we hope is going to help us, because if we compare, this is just looking into the future of the next data, we have a really dry 25 compared to 24, which was really challenging. We are expecting quite a dramatic impact in the microbiome, in the sense that it's going to be, some of our samples, they're going to have a quite dormant microbiome, so perhaps it's going to be very difficult to link all the functional parameters with what the microbes are actually doing. So we will see, but I just wanted to draw your attention. There was really a decrease in rainfall, quite dramatic, like I say, particularly during the season when wheat is growing. And that was accompanied as well with an increase. So according to several sources, like the Met Office, we have one of the warmest summer in record last year. So let's see, for the next seminar, I will have to apply that. So now this is a bit of more heavy statistics. It's actually the linear modeling. It's I have done quite a rigorous, quite an aggressive filtering of the data, because there is two limitations to this. One is obviously actually the effects of the linking microbiome with other things might not always be a linear impact. And also there is so many cakes. The list is that we lose a lot of statistical power because we have 200 samples and also kind of 100 or so genes that we are looking at. So that's going to limit sometimes our capacity to actually detect a significant impact. But nevertheless, after doing I think it's a good start to screen the data. So then we can go into other approaches that are perhaps more robust or more adequate to process this data, because at the end of the day we want to find links. It's like which genes are being linked to nitrification rate, which genes get modified for the management, which genes are more related to soil properties, which genes are more related to higher arbuscular mycorrhiza colonization, etc. So in this particular case, I have separated because, like I say, statistically, it's not possible. It was not possible with this modeling to put everything together. So I thought it was a bit more honest to separate it. So what we can see is like impact of different basic organic matter pH, water holding capacity, and the nitrification rates on the variables that we measure with a CT scan, and in the variables that we get from the microscopy analysis for the AMFs. And this is the list. So the list of functions, well, modules actually, that I'm displaying here that get significantly correlated with this PNR, with these parameters or with this parameter. I selected them from the ones that are significant according to the analysis, and also because they have a more immediate ecological meaning. Because at the end of the day, we want to learn and try to explain to the root rangers what is actually what's the impact of your management on the microbiome functioning. So don't worry, there is so much point in really going one by one. Some of them might ring a bell to you, but I have made a summary in a couple of slides where I'm distilling the main meaning of this. I just wanted to explain you a bit how it works. So same approach, you will see the same slide for the casein, remember, for the carbohydrate turnover. And again, we will have the most significant ecological, most significant from the analysis, and then most significant for ecological mean. These things like cellulase or CNase or lysozyme might ring more a bell, but obviously you want to see them in a context. So a lot of readings, so I'm just going to highlight very quickly some things. And I invite you to, since you're going to have access to the slides and you're going to have access to the recording, feel free to dig a bit more into that. But basically, we have some KEGG modules that are really linked to the nitrogen nitrification rate, the AFM, and the CT, as you have seen many of them, including things like metal transport models, which actually they are related to the nitrification rate because the nitrifiers need this exchange of, they need some metals to make their enzymes work as a cofactor. So this transport is very important for them. So that's why if this gets affected, it will affect the nitrification. And same with this cytochrome. So we are talking about all the machinery and all the things that the plants, that the microbes need to do this nitrification rate. We didn't find as much, although we have some hints of some links between this functional change in the cakes and the solid structure, but nothing really that solid. There was more response from the AMF. They seem to be much better predictors, and the aromatic compound degradation in the soil seems to positively associate with the metrics. And obviously organic matter, which is very simple from the soil parameters, very basic, but very important, is one that is really strongly correlated. And same thing, so cell degrading enzymes were also important for AMF, another cutinase, and there was more association on caseins. They are comparatively weak, compared to the other ones, but there is still a hint that we with more repetition, with more replicates, we probably will have a bit more. We could be much finding some more positive correlations. Again, organic matter is also. And the last one of the last heavy slides, if you want, I was telling about the limitations of running a linear model, mixed models. So this kind of structural equation analysis, it's much more powerful in terms of finding these links. Don't get too ditzy looking at all the arrows going up and down. Remember, we have management, soil properties, some of the functions I have highlighted here, like nitrogen fixation, denitrification, carbon turnover, and some of the basic ones, and then how all of these will reflect, so how these changes reflect in the actual things that we measure. So some things will, here you have a code of what the different things meaning. So the little numbers will give you when there is an actual significant impact. So there is significant link in between these two, for instance, between management and nitrification. So you will see the arrows. Here we only see two because obviously all of this is comparing to the conventional management as a reference. Blue is for positive, red is for negative impact, and the width of the arrow will tell you whether the effect is really bigger. So the thinner it gets is a much weaker effect. Now let's see. It confirms some of the things that we already saw in a more basic way with the linear modeling. So we see an enhancement or a potential enhancement of AMF colonization for both organic and regenerative compared to conventional. Obviously, pH and organic matter are going to be strong drivers. We see some improvement of soil structure by having more pore networks. I was mentioning pores in the earlier on because obviously they create these places where water can be stored and they create microhabitats for the microbes. And nitrogen fixation seems to be, according to this, to our results, could be, I always prefer to put the might be, so it looks like, could be improved, could be linked to improvement of pore connectivity. So overall, we do see management effect, although a lot is driven by the soil characteristics, particularly things like pH. But there is a hope or there is a hint to positive outcome of that there could be a suppression of this nitrification and enhancing beneficial fungi. I want to say we are working quite on finalizing all this analysis. So hopefully you will have some manuscript available in bioRxiv, and hopefully in the next few weeks where you will see that you can read more in detail all the story that is building from these results. And with that, I just want to finish my talk linking. So because of the nature of this talk, is a bit back to forth. So Truth is running 24, 25, and 26. The Wheat-Roots so all the experimental field trials were run during 22, 23, and 24. So it was sometime during 23, at some point that Tom got an interest in all the methodological approach and all the microbiome work that I was doing at Joninas. He came to many of my talks, and I was really thrilled that he went ahead with the Truth project to have us as partners for two main reasons. So we are running, like I said, all the microbiome aspects of the project, but also because for this year, the root ranges. So for the first 2 years, they have been growing the wheat cultivar of their choice that one from the recommended list. This year they are going to be growing different wheat varieties that I'm going to present in a second that have been selected from the results of the Wheat-Roots. So, like Sylvie mentioned earlier on, this was a large international collaboration with the field trials from China to South Africa to different countries in Europe. And we wanted to look how root. So we examined a panel of 20 different wheat genotypes, we call them, or varieties if you prefer, but I prefer the term genotype. And we were characterizing the root architecture. and at the same time, for same samples, measuring aggregation. For some of the samples, we did also the tomography, in collaboration with Nottingham as well. And then we also wanted to see the impact of potential exudates that different genotypes will have in the microbiome composition and functions. So this was a bit more limited because we were doing 16S and ITS, so it's not as powerful as running shotgun, which allows a much more a prediction of functions that we can trust a bit more. So for the last for my second and last year, for my field trial in UK, I actually used shotgun with Novogen. I put the same samples in the pack of the truth because I actually wanted to have a more comparison close to the UK soils. So just for any of you that is not familiar with the Watkins collection, it's a large collection of wheat genotypes with a fantastic genetic diversity that we keep in the germplasm at Unines. Anyone that is interested, you can contact them or contact me about the potential of this old wheat, all this historical wheat, to bring new traits into modern varieties. And well, one thing that is interesting is that all the sequence from all this wheat was completed a long time ago. It's been a kind of a 10-year work. And they found that some of them group in these two groups are closer to the modern varieties that we grow in the field these days, while all the rest are much more different genetically. And that translates into carrying many more genes with potential beneficial traits that we could breed back into modern cultivars. And one of my very early studies that is published, if you want to, well, it's available in bioRxiv if you want to have a look more in detail. We found that the microbiome composition, this is just phylogenetic diversity, actually mirrors in difference the genetic diversity above ground. So as you can see, AG2 and AG5 are the only ones that are not significantly different in phylogenetic diversity of the microbiome. So that was my first study. Like I say, the Wheat-Microbe was measuring these 20 genotypes, 2 nitrogen fertilizer. So as you can see, we're using very low inputs because these cultivars are not, they predate modern breeding and the inputs of fertilizer, so they are not used. So we give them a minimum. And then we have a 96, which is still lower than farm standards. And we ran, in these conditions, we ran the parallel field trials for 2 years in 7 countries, collecting, remember the compartments, Rhizosphere samples and their soil samples as control. And just one sneak peek, I'm working on thousands of samples that we have created from this. You are familiar now with this kind of plot, I hope. Again, we are just looking at, in this case, just the kegs. And one of the main outcomes that I have seen is different types of the kind of kegs for cycling, kegs for energy, kegs for transport. Some of them we have not yet enough statistical power. I'm hoping to improve it or to advance a bit more the analysis in the future. But we do see for some very general modules, we actually see an impact on the nitrogen application, the amount of nitrogen that has been applied. So now from my results on this, 4 of them have been selected. So some root ranges are growing 2 of them, the bred ones, and some of them they are growing the durum lines. Because the bred ones have potentially could decrease nitrification rate with the exudates you put in the soil, while the durum one, we are selecting them more for this impact in the soil structure. So just a little message to leave it in a happy note. Regenerative agriculture is very much in transition. And one thing we are trying to bring into the equation of this transition, like I say, is many different things can be called regenerative approaches. Sometimes you combine 2, sometimes you combine more. But the thing that we want to do is actually have some root traits being into this equation, bring some traits from roots into the new cultivars that can also be considered a part of, because that will translate in a better soil structure and that would also translate into decreased inputs, which is very much part of regenerative agriculture. And with that, I just want to say thank you, and I'm happy to take questions.
Thank you. It was really nice talk, Maria. Thank you very much to give, this talk during. And now, we have a few questions. So, let me check. So, the first question is, from Alexander Anderson, and asking, I'm currently researching RNA viruses from soil samples, some of which originates from agricultural soil, and could that perhaps be of relevance to your research as well?
It would very much be, because obviously I already presented quite a lot. We do have another Innovate project with a company that is working with phages. So this is a thing in the side, if you want. we have been working for 3 years. We don't look specifically, I haven't looked at RNA yet. That's something for future projects, actually, because we are closing with projects at the minute. RNA is very much something I'm interested in, and Selvi is offering help. Yes, I'm very interested in viruses. They have a tremendous, we have seen practices impact virus load in soil. It's still a very niche area of research, and it's quite difficult. We are very, as you probably know, we are very limited by databases, and doing it in pure soil is really difficult because there's so much noise. But yeah, I'm definitely super interested. I'm particularly interested in phages that infect nitrifiers, Kia, and so that's one of the things I want to pursue in the future. Thank you.
Okay, good. Thank you. So, another question is from Gabriel, and asking, in terms of sampling and sequencing for whole genome sequencing, how many biological replicates per soil, meaning how many FASTQ files and how much depth you require to ensure all biodiversity is well captured?
So soil scientist, there is never enough samples. So for the truth, we have, since we didn't find any difference between, I think I could show you actually the actual numbers. So each farm collected 22 samples, 11 before the fertilizer and 11 after the fertilizer. And there was no difference, so we could kind of merge them. Yes, no, that's a number of farms. What we are a bit poor in this first year, that's why we want to pile the 2 years together when the data becomes available, is the number of farms and the number of farms per soil. But in terms of sequencing, we have we saw like 12 replicates for the Rhizosphere soil, which is per sample per farm. And they are taken from composites. There is a video about how we sample somewhere. Boffin posted a video on YouTube about for the training of the farmers. So basically, we have kind of this W, and in each point of the farm that they take a sample, they will also take several plants, or they will take a composite. So each location replicate is also a kind of biological replicate. So it's reasonable. So that means, we have from this study was over 200 samples. So if we have the fast and reverse, it's 400. So a lot of fast.
Yeah.
We have a high performance computer at the research park. Otherwise, it would be. So we can do the analysis with the pipelines over 2-3 days, sometimes less. But otherwise, it will take. The data is very heavy. Self-sense with really rich data.
Yeah, exactly. Actually, if you don't mind, I like to add a few things about especially FASTQ files. Yes, we need to calculate it, but normally, we are using parents 150, and which means the parent is we are delivering the 2 fascicles for per sample, and which means yes, you need to calculate it based on the sample number.
Yeah, so
Thank you.
we do it by lanes, right?
Yeah, we do lanes, and we use a flow cell of Nova 6X plus, which is 25B, and each lane has 1 terabyte data output, which means about 3.3 billion, and the total one is 6 billion reads. So yeah, each time it's necessary to calculate it, to be honest. It is not, there is really exact response for this kind of technical questions.
Yeah, well, it has to be also the sequencing depth has to also be taken into account, because the lane gives you
Yeah.
the amount of data that you can generate. So then you have to kind
Mm-hmm.
of balance. Remember, we have plenty of conversations about that over mail, of how to
Yeah,
balance the sequencing
definitely.
depth with the sequencing depth for soil samples. So obviously we want to have as much depth as possible to do the assemblies, but with more depth sometimes comes a bit more of noise too, and a lot of chunks
Yeah,
that are. So we try to find a balance in between having good data and.
definitely. Thank you very much, Maria. And we have a question from Javier, and asking, yeah, very good presentation and incredible project. So do you think that big microbial variation could happen if the fields were treated with different nitrogen fertilizer, like ammonium, nitrate, or urea, for example?
Uh, to be honest, each farmer was applying. I mean, we have a data log with all the different fertilizers that because each farmer pretty much is using a different combo. Obviously, the organics, one is using compost, another one is using so no chemical fertilizer. So for this first year in the 24, we didn't have enough. There's too much variability in what they are. So I would say that what you can infer from our results is that if we have limited difference between the actual management as a big organic conventional and so on, I would not expect that much impact. But most people just use a combination of ammonia and nitrate. Now, I do expect that it makes that's something that I want to do for the future, because for the wheat root, we only apply ammonia, while
Mm-hmm.
most farmers apply, because obviously it's for, I'm sorry, more in the sense of how much of the ammonium transform into nitrate, which was our research question. So there has to be an impact in the way that obviously but surprisingly, I don't find as much impact by giving a lot of ammonium in the microbes than I expect. I expected a really dramatic impact, and none of the countries has been. Sometimes some of the countries in the study and some of the years we have just put all the before and after fertilizer together. But yeah, you would expect an impact in the different fertilizer. Basically, it has to be combined with all the other things: the soil type, the soil characteristics, the rain. So it's difficult to answer
Yeah,
one isolated factor. You need to put it
yeah,
together in the equation, in the big equation with everything else.
great. And we have so many questions. Sorry, we just, reach our end of the session. Just,
Ooh, that's a lot of
just I
questions.
think I am, yeah, maybe it would be the last question, but don't worry, we will answer to all of the questions after the webinar. So question is from Vincenzo, and yeah, hello Maria, super interesting presentation. I have a question regarding the microbiome network. Which taxonomic level have you used in this work, and which level do you recommend to use for such analysis?
Well, the analysis, I mean, with the shotgun, we go to species. Uh, one problem is that the databases for soils are limited. So we can get this species resolution. And if you look at my taxonomy tables, they are all. The problem is you end up with a lot of unidentified ones. Then you have to go back. So normally, for the purposes of, for the main statistical analysis, we just use the CSVs. But if I want to present a taxonomy table to the farmers, for instance, or for a presentation, or if I want to present something for a paper in a supplementary material, I will have to probably restrict myself to the genomes, because despite the fact that we have the resolution, the databases at the end of this pipeline are really limited in identification of species. So
Yeah.
it depends what your question is. Yes, we can go to species resolution, but what does it mean if we cannot actually assign name or
Okay.
So that's why also I like doing the kegs, because at least we have chunks of genes, and then you can see, well, maybe you don't know the name yet, but you know there is a gene there. And that's more important, perhaps, than just looking at the names.
Cool. Thank you very much. And yeah, thank you for all your time today. And we hope you enjoyed the webinar and found it useful. So yeah, thanks also for all your questions. Yeah, as I mentioned, if we haven't managed to answer your question live, and we will get back to you via email. So next week with the answer. So also we will email you a link to the webinar recording and the slide deck. So yeah, you can expect to receive the week after. When the webinar finishes, a survey will pop up, and we appreciate you taking time to complete that. This really has, as it helps ensure we make future webinars even more relevant to you and your research goals. And yeah, there will be a link in the follow-up email for you to view a certificate of attest attendance if you need the one. Yeah, this can be also printed from your browser window. In meantime, you can follow us on Twitter, LinkedIn, Instagram. So to keep up to date with future webinars, offers, and news, and we look forward to seeing you all the next webinar. Thank you very much, and Maria again to be with us together as well with your amazing research. And thank you making us your research partner during the sequencing. Yeah, we were really happy during this the research with you.
Thank you, everybody, and particularly to you guys at Norwegian. You're fantastic.
Yeah. Thank you. Have a lovely day. Bye-bye.
Bye-bye.